I always think it's great to ask people what kind of advice they have for young professionals who want to sort of follow in your footsteps and may want to make an impact in their research careers.
Well, I'll provide my 2 cents. Go to the old books. So many of the deepest ideas are already there. They're hidden in plain sight, in a way, and then just waiting to be rediscovered in a new context. Oftentimes, the new ideas we got excited about are old ideas that finally found the right problem.
Hello, everyone, and welcome to the Biophotonics Discovery Podcast. Whether you're a seasoned researcher, a student just starting out, or you just love a good story about how light is used to solve big problems, this podcast is for you. We're making the discoveries in biophotonics accessible one episode at a time. Well, welcome, everyone. Today, we have a very special episode of the Biophotonics Discovery Podcast. My name is Darren Roblyer, and today I am joined by Travis Sawyer, an associate editor for the journal. Together, we will be having a conversation with the recipient of the journal's inaugural Impact of the Year Award.
Thanks so much, Darren. I'm excited to join you today for the interview.
Great to have you, Travis. We are thrilled to welcome the Impact of the Year Award winner, Dr. Guosong Hong. Guosong is an assistant professor of material science at Stanford University, where his lab aims to harness fundamental principles of material science, physics, and chemistry to solve pressing challenges in biology and medicine. Welcome, Guosong.
Thank you. Thank you, Darren and Travis. I'm glad to be here.
Thanks so much for joining us, Guosong, and congratulations on the award. So, to give a little bit of background to the audience, the Impact of the Year Award is an annual award that was established last year by a committee composed of biophotonics discovery editorial members, including Darren and myself. The intent of the award is to recognize recent and specific contributions that have had a meaningful impact on the field of biophotonics. Dr. Guosong Hong was recognized for his pioneering work in optical clearing, where he showed the incredible ability to achieve optical transparency in live animals, which was presented at SPIE Photonics West in 2025 and then subsequently published in Science. And I think most impressively, since then, this discovery has already been reproduced and utilized in over 20 independent publications, which really illustrates just how impactful and wide-ranging it really is.
So to start things off, Guosong, we'd love to hear a little bit about your scientific journey. Could you tell us a little bit about your training and some of the main ideas you've been thinking about and working on, and then how you ended up focusing on this specific work, the optical clearing work?
First of all, I'm really honored to have received the inaugural Impact of the Year Award. So, my journey into science and especially biophotonics started during my PhD at Stanford. I was working on in vivo fluorescent imaging in the pretty unconventional wavelength range, which is what we now call the second infrared or shortwave infrared window, around 1 to 2 microns. What is really special about that window is that light scatters much less than in the visible spectrum. So you can see deeper into the tissue in this particular window. As a chemistry student, I was making carbon nanotubes and quantum dots that emit in this special window, functionalize them to make them water soluble and biocompatible, and injecting them into mice, imaging through intact tissues. One real eureka moment was realizing that if we chose just the right wavelengths around 1.3 to 1.4 microns, we could actually see brain vessels and even blood flow in the brain in live animals in a completely non-invasive manner without opening the skin or the skull. That was a moment where I felt, okay, light can do something really powerful here. So then I took a detour during my postdoc at Harvard, where I worked on electronic tools for brain recording. But when I started my lab back at Stanford, I found myself drawn back to optics again. There's just something uniquely compelling about light. Then the pandemic hit, and during the lockdown, my team and I went back to some basics. We spent a lot of time reading and discussing fundamental optics on Zoom. One book we studied closely was titled Absorption and Scattering of Light by Small Particles by Bohren and Huffman. That reading and discussion led to another key insight. Scattering and absorption of biological tissues aren't independent. Through the chronic radiation, changing absorption at one wavelength actually changes refractive index and therefore scattering at another wavelength. That realization shifted our thinking. Instead of trying to remove scattering, we asked, could we engineer it away by tuning absorption instead? That idea ultimately led to our collaboration with the Huffman lab at Stanford and to using absorbing molecules to make scattering media, including living biological tissue, become more transparent.
Awesome. Well, thank you so much, Guosong. That's a great story. And I particularly like how you discussed how an obstacle like the pandemic actually led to an opportunity that you were able to take with your lab to go back to the basics. And
Mhm.
so I think I can speak for a lot of us in biophotonics saying that we were really excited about how your discovery and optical clearing could impact our own work. But I'd like to hear from your perspectives. What are some of the biggest ways you see this discovery advancing biophotonics as well as other fields?
Thank you for the question. I think what's really exciting is how broadly this approach can be applied, both within biophotonics and beyond. Optical methods are foundational in biology and medicine. They let us image cells, control neural activity, and perform light based therapies with incredible precision in both space and time. But they all run into the same fundamental barrier. Biological tissue is inherently opaque. What our approach does is to address that limitation at its root. By reducing scattering, we can potentially enable us to see deeper, sharper, and more comprehensively in three-dimensional living organisms. Another key point is that this isn't tied to one specific molecule. It's really a general physical principle. We and many other labs have shown that many different absorbing molecules can be repurposed as clearing agents, some of which are already FDA approved, like fluorescein and indocyanine green. And it doesn't stop at biology. Anytime scattering is a limitation, which is true across many optical and photonic technologies, the same idea could be applied to improve performance.
Yeah, really interesting, Guosong. While you were talking, I was also looking at my bookshelf, try to find my copy of Bohren and Huffman. It's awesome that you've gotten new innovation and insights out of that, classic text. So let me ask you something about this work because, one of the things that's true, I think, for a lot of us is that not every challenge we have to overcome gets clearly stated in a manuscript, right? There's a whole lot of things you probably fail at before you're successful. And I'm wondering if there's any specific challenges or things you had to overcome that you want to tell us about, that aren't so clearly delineated in the paper.
Yeah, absolutely. Well, first of all, I actually keep a copy of that book on my desk all the time because that's my frequent go-to book. Whenever I need to check something, I read that book. So it's really a great book. Now, getting to a question on the challenge that isn't captured in the publication. One challenge we encountered was identifying the right absorbing molecules. At first, the Cremer's chronic framework made it seem like many candidates should work. But we quickly learned that most good absorbers or good absorbing molecules or dye molecules are actually too good. They carry a long absorption tail into the visible and sometimes even all the way into the near infrared. So absorption ends up dominating over scattering reduction. In the end, only a handful of molecules pass the test. These molecules need to absorb strongly, but then the absorption needs to disappear very quickly beyond where the peak ends in a longer wavelength. So tartrazine is actually one of these molecules, and as it turns out, in some of the recent works by others following the same framework, fluorescein and indocyanine green also meet this requirement. And I believe there are many other molecules that can do the same thing by satisfying this requirement.
That's really interesting. If I could ask a follow-up question on this, when trying to evaluate candidate molecules, do you do the characterization measurements yourself, or are you referencing other literature that kind of shows this data? How is that done to kind of figure out which ones are the best candidates?
Yeah, that's a great follow-up question. Actually, we are doing that in both ways. There are a lot of databases we are resorting to get the information, specifically spectral information that has been collected by other researchers. And this enables us to quickly screen many molecules to identify those that can satisfy the requirement. In the meantime, we're also carrying out measurements ourselves in our lab on some of the new molecules, either synthesized by ourselves, or some of these molecules are actually interesting biomolecules that we purified from biological tissues that have been hypothesized to give rise to some interesting optical properties of these biological tissues. And then we based on this knowledge, based on the measurements we're doing in our lab, we identify those that could potentially be biocompatible while also achieving transparency.
That's great. Thank you. Yeah, and I like kind of your comment on how existing databases could, provide some suggestions kind of on what things to look into, because I think it speaks to the value of open science and kind of making data available.
Exactly.
So now that you've made this big discovery and, it's obviously getting a lot of attention, what's next for you in this line of research?
We're currently working with our collaborators. So some of them are Stanford dermatologists to apply this approach to human skin with the goal of making skin cancer diagnosis less invasive. We're also exploring applications in the eye, aiming to restore transparency and thus vision in opaque cornea and lens tissues in the context of, let's say, corneal burn or cataract. Beyond these applications, we're also pursuing a more fundamental question. What molecules can provide the strongest absorption, thus requiring the lowest concentration to achieve transparency? Answering this question turns out to touch on the quantum mechanics of light molecule interactions. And along the way, we have uncovered some surprisingly fun insights, such as what is the physical limit of the absorption cross section per molecule, and how can we get closer to that physical limit by synthesizing some new dye molecules.
Oh, that's really interesting, Guosong. And it seems like there's plenty of work left to be done in this area. So that's exciting.
Yeah,
But we know your lab, probably works on a lot of other things. Can you tell us anything about other projects your lab is currently working on?
certainly. We work on another project that I call quote unquote seeing the sound. We found materials that can convert focused ultrasound into light. So what really motivates this work is that currently people rely on optical fibers to deliver light deep into the body. For example, you can use focused light to generate molecules called radicals that kill cancer cells, bacteria, and viruses. You can also use light to control brain activity via optogenetics. But all these methods require the invasive implantation of fiber optics. So, in contrast, ultrasound can naturally penetrate much deeper than light. So, you could focus ultrasound very deep in the body and then couple ultrasound with some circulation delivered transducers. These are nanotransducers that can convert ultrasound into local light emission and then produce this light illumination pattern at any location or depth inside the body in a non-invasive way. And then we use this approach to demonstrate this ultrasound scanning in vivo light source can be used for non-invasive optogenetics, neuroscience studies, as well as things such as ultrasound-enabled gene editing by coupling with a photo-switchable CRISPR-Cas9 system.
Help me understand. Do you have to implant some material in the tissue that
No,
is?
actually,
No?
you don't. The nice thing about this is you just need to inject some of these materials that are nanoparticles into the bloodstream, and then these particles will travel with the circulation. These particles get recharged near the skin when they are passing through the superficial vessels, and you can apply UV light to shine on the skin to get absorbed by the material to recharge this material. And then these particles flow with the blood until they pass through the focus of the ultrasound, which is usually much deeper than the superficial vessels. And then at the focus of the ultrasound, they emit light in response. And this light emission is very focal in the region of the ultrasound, thus producing a very small light source wherever the ultrasound is applied. After losing the energy due to light emission, this material still stays in the circulation that would pass through the superficial vessels again, get recharged, and then get discharged again by ultrasound. And this process just keeps going on as long as we have this material circulating in the body. So this is basically a way to leverage circulation to turn blood circulation into an almost like an optical flow battery in a way. And then we use that to produce light emission based on the scanning motion of the focused ultrasound.
Wow. And what's an example of a material that has this property?
Yes, right. So the material is called mechanoluminescent materials. And in the past, this material has mainly been used in ceramics, in the some really bulky ceramic form. But because we are also doing some chemistry on the material, we could turn them into really small, water soluble, and biocompatible nanoparticles that can be delivered in vivo.
And what kind of emission wavelengths can you achieve with these?
Yeah, actually, we're working primarily with blue emitting mechanoluminescent materials
Uh-huh.
with the wavelengths of 470 and 490. These are the two particular wavelengths of the materials we have worked on. But there are many other materials that emit all the way to 650 nanometers. And then we have also demonstrated that these materials can be made into the nanoscopic nanomaterial form and enable a wavelength and color tunable ultrasound responsive light source.
Hmm. But the short wavelengths will be useful for optogenetics, right? Don't a lot of those systems use blue light?
Exactly, that's right.
Yeah.
Primarily for excitatory options. For inhibitory options, people typically use redshifted excitation, redshifted light.
That's
Okay.
yeah, that's really interesting. I feel like it's sort of inverting the photoacoustic paradigm.
Yeah,
Um,
that's right. This is like a acousto-optical.
yeah. No,
Yeah.
fascinating. Thank you for sharing. And, I'd known about materials that have that kind of relationship between mechanical stress and strain and
Mhm.
generating light, but I've never actually seen such a great application of them. And so that's awesome to learn about. I had actually a quick question on an earlier comment you made about your optical clearing work being applied to restoring vision with like patients who may have cataracts. And just some background. I've done a little bit of work in like intraocular lens development, which generally the cataract, the physician goes in, ablates the old lens, removes it, and then reinserts a new IOL.
Yeah.
I'm curious with this kind of idea of restoring transparency, how permanent is the administration of these compounds or these molecules? Is it something that could last for years?
Yeah, that's actually an excellent question. So currently, the cataract surgery, it takes about 30 minutes, I believe, in practice, which is not so burdensome.
Yeah.
But then for corneal replacement, right? So whenever there's significant damage in the corneal tissue, in the stroma of the cornea, the
Right.
resulting opacity requires transplantation. And for people, depending on where the patients are in the ward,
Mhm.
the wait time for donor can range from a few months to a few decades. So now the goal is to use a eye drop in a way to restore vision. Currently, our demonstration is still just a transient. So with an eye drop, we have demonstrated we can restore vision up to 30 minutes to about 1 hour. And after that, this goes back. And this demonstration has been demonstrated in live rabbits, not in humans yet. But the one possibility is to have these molecules cross-linking with the extracellular matrix in the corneal tissue, thus making this more permanent than transient. So that is the one possible route to get this to be a long-lasting effect rather than a 30 minutes transient effect.
That's really interesting. Yeah, thank you for sharing. I think the cornea is a really interesting organ because it's so accessible, and I feel like that's a
Right.
fantastic way to address both the permanence and you're totally right, spot on that corneal transplants are really hard to do. Just
Right.
give it, you don't have many corneas lying around. So taking a little bit of a detour now away from the science, I always think it's great to ask people what kind of advice they have for young professionals who want to sort of follow in your footsteps and may want to make an impact in their research careers.
Well, I'll provide my 2 cents. Go to the old books. So many of the deepest ideas are already there. They're hidden in plain sight, in a way, and then just waiting to be rediscovered in a new context. Oftentimes, the new ideas we got excited about are old ideas that finally found the right problem.
That's good advice, Guosong. And maybe now with LLMs, we can use those LLMs to extract some ideas from those old
Yeah,
books.
exactly.
Yeah, exactly. All right. Well, it's been great to learn more about your work, Guosong. Thanks for joining us and congratulations again on your award. We look forward to seeing your next discovery. It's clear there's going to be many more in the future. And I just want listeners to remember that we'll be seeking nomination for next year's Impact of the Year award, probably in the early fall. So stay tuned for that. Thank you, Guosong and Travis.
Thanks,
Thank,
Darren. Thanks, Song, and congrats again.
thank you, Darren and Travis. It's been a great pleasure and honor to be here.
If you enjoyed this episode, please subscribe, leave us a review, and share it with your colleagues. Your support helps us shine a light on the latest in biophotonics research. Stay curious, and we'll see you next time.